Hi everyone, welcome to this session. We will talk about disaccharides, which is a subtopic of carbohydrates, which is in turn the subtopic of biochemistry. So disaccharides are made from two monosaccharides. For here, we're going to look at a specific example, which is maltose, and maltose is made from a glycosidic bond or a linkage between two glucose molecules. And this process you might be familiar with is called condensation, which removes a water molecule. So inspecting it closely, we will see that the hemiacetal anomeric carbon is being attacked by an alcohol from another glucose. Recall that in our last session on monosaccharides, we learned that the linear form of a monosaccharide can be converted to the cyclical form of a monosaccharide by attacking the aldehyde carbon with an alcohol that's from the same molecule. So therefore, we can call the product an intramolecular hemiacetal. And now we can complete this process by attacking this hemiacetal carbon with another alcohol, though this time the alcohol doesn't come from the same molecule. It comes from another molecule, another glucose in this case, finally forming an acetyl carbon. So if condensation removes a water molecule in this process, hydrolysis inserts a water molecule in this process to break the glycosidic bond. In terms of representation of disaccharides, I would like to point out that the wavy lines here on the hemiacetal carbon of the glucose on the right side can be either alpha or beta, because this glucose will still be able to convert between its cyclic form and its linear form. And thus, if it's alpha, it can be converted to a beta form, and if it's beta, it can be converted back. So it could be alpha or it could be beta. So thus the wavy lines here are trying to depict this sort of ambiguity, and we call this mutarotation. Though it wouldn't be meaningful if we apply mutarotation to the acetal carbon, since this previously hemiacetal anomeric carbon now is locked into place by the glycosidic bond, and the glucose monosaccharide on the left won't be able to convert between its cyclic form and its linear form. So it won't be able to turn linear. So we can identify it here, specifically in this case, as alpha, and say for sure that it's alpha. So looking at the hemiacetal group on the right even more closely, sugars that have an aldehyde group can reduce copper 2+ ions into copper plus ions, and thus oxidize the aldehyde group to carboxylic acid. So basically, reduction means the gaining of electrons, and in this case, copper ions are gaining electrons, and aldehyde groups are losing electrons by through the process of the aldehyde carbon being connected to more oxygen, which becomes carboxylic acid. So sugars that can do this needs to have an aldehyde group or a ketone group that can tautomerize to an aldehyde group. So we call sugars that can do this redox reaction with copper 2+ ions reducing sugars. And since the anomeric carbon here can convert back to the linear form and thus have an aldehyde group which will which can form redox reactions with copper ions. We call this the reducing end, and thus maltose is a reducing sugar because it is able to perform this reaction with copper 2+ ions. And this will be important later on in this session. So how do we name disaccharides? First of all, we have to figure out the configuration of the anomeric carbon specifically for the glucose on the left, because it cannot convert between its alpha and beta configurations anymore, since, as I said before, it's locked into place by the glycosidic bond. And from the last session, we will know that because the O or the OR group is on the opposite side of the plane than the 6th carbon, we call this the alpha configuration. Secondly, we have to identify the enantiomer of the monosaccharide, and in this case, it would be glucose in the D form. Thirdly, we want to see if the thing is basically a hexagon or a pentagon. If it's a hexagon, it looks like purine. If it's a pentagon, then it will look like furan. So here it's a hexagon. That's why we add purine to its ending. And we do the same process. We identify the enantiomer of the monosaccharide on the right and its shape. And the only difference is that we don't have to figure out the configuration of the anomeric carbon because, as I said before, it can convert between alpha and beta forms since it's not locked into place by a glycosidic bond. Lastly, we want to know the 2 carbons on either side of the glycosidic bond. So here, it's the first carbon that's bonding through the glycosidic bond with the 4th carbon of the glucose on the right. Therefore, it's a 1, 2, 4 sort of formation, I shall say. And we also have a shorthand for this unnecessarily long name. Maybe not unnecessarily, but yeah, anyways, which is GLC, which stands for glucose. And there's 2 glucose together, and the alpha dictates the configuration of the anomeric carbon on the glucose on the left. And then 1, 2, 4 again shows that it's the first carbon of the left glucose on the left side of the glycosidic bond, and the 4th carbon on the right glucose on the right side of the glycosidic bond. So let's look at some examples of disaccharides. The first example will be lactose, which gives milk its sweetness, and it's formed it's formed from a galactose and a glucose, and so the lactose in its beta form will have the galactose in its beta form. And then again, it is a reducing sugar because, it is a reducing sugar just like maltose because its anomeric carbon on the right, can form redox reactions, can perform redox reactions with copper. So since it has this reducing end, we call it a reducing sugar. And just something else for you to know, the enzyme lactase in the small intestine, which is not present in lactose intolerant people, cleaves the glycosidic bond. And the monosaccharides, which is galactose and glucose in this case, can be absorbed in the small intestine, while disaccharides cannot. So for people who are lactose intolerant, since the lactose cannot be broken and absorbed in the small intestine, it goes to the large intestine, which makes the lumen of the large intestine more hypotonic because the lactose will decrease the amount of free water molecules in the lumen, and thus it decreases the large intestine's ability to retain water and to absorb water from its lumen, and thus it leads to watery stools. Also, fermentation of lactose by intestinal bacteria can lead to cramps and gas. So next up we have sucrose, which is which is table sugar, and it's formed from glucose and fructose. It is to note that it is a non-reducing sugar because both anomeric carbons from each monosaccharide participates in this glycosidic bonding. And sucrose is made by plants, not animals, and since it is non-reducing, it is stable since it cannot be oxidized, or it cannot form redox reactions with other things. It is useful for storage and transport. In fact, many plants uses sucrose to transport energy from leaves to other parts of it. So then we have trehalose. It is another disaccharide of glucose, though it is differing from maltose in terms of having the glycosidic bond between the two anomeric carbons, and thus again it is a non-reducing sugar. It is present in the hemolymph of insects, probably for the similar reasons as sucrose. It is used for energy storage, and it also might function as antifreeze in some invertebrates. Thank you so much for watching this video. You should anticipate the next session, which will be on polysaccharides.